on Thursday, finished chatting about microscopy. And we mentioned among three major categories, major characteristics of a microscope, one characteristic is contrast. Right? That's important. And we talked that samples that we look at under the microscope, they're mostly made of water. So contrast is kind of lousy. You could, when we did a wet mount, you could see that, you know, it's far from perfect, those images, just natural cause. So wet mounts, of course, are good, but we want often to stain the specimens. Another problem that, well, not a problem, if you have a buttload of microbial cells on your specimen, you don't really care if they're moving or not. Okay? If you have taken 1510 or 1200 here, you may have done a microscopic study of water. Have you? Some of you? And if you look there, there's some little creatures, protozoa, mostly paramecium, that swim around. And to trace one is really a task because they fast. Okay? Same goes for bacterial cells. They can move. And in many cases, you want to stop them before you observe them, so they don't go anywhere. Does that make sense? So for this, you fix your specimen. You can fix it by chemicals, which we aren't doing, or you can fix it by temperature. The fixation by heating, as shown here, is probably better because you can exactly time the fixation process. You can say we place it at 50 degrees Celsius for say 2 minutes and it's fixed. But we're going to do fixation over the flame, and it's fairly simple. If you have a flame going on and you have a slide, okay, sorry, so what we're going to do, we're going to use flame. Flame goes, here's your slide. You do across the flame, like across the very top of the flame. 1, 2, 3. That's how fast it should go. 1, 2, 3. Your bacteria are dead. It will take some time to get them just dead, because in many cases they pretty much explode. If you overexpose them to the flame, it's going to be a mess. They're all going to be dead, they're going to disrupt, it's going to be a problem. But usually it works pretty well. Okay? Now, when you fix, then you can stain. There are two big types of stains. The simple stains and differential stains. Both of them serve pretty much the same purpose, to increase contrast. Okay? Simple stains do not distinguish between different types of microbes. These are two examples of simple stains. Okay, they somewhat different, but they're simple stains, which means if you take gram-positive rods and gram-negative rods and put them on the same specimen and stain them with, say, crystal violet, you won't be able to tell them apart. Does that make sense? So crystal violet will stain all cells dark blue violet. Does that make sense? That's your simple stain. Simple stains or differential stain will let you to tell apart different types of cells. We'll get to that. Simple stains can be positive. In this case, the molecule of a dye, like crystal violet molecule, is charged positively, it will bind to the surface of the cell, which is generally charged negatively, okay? And it will stain the cell itself. You can see the chains of Bacillus anthracis here stained the violet color, right? Because stain attaches to them. Background remains not uncolorized, right? Negative stain, which you can see here, stains background. Things like nigrosin, okay, or India ink, okay. They stain background and they don't attach to the cell. Bacterial cell essentially repels them because they're negatively charged, and so is the surface of the cell. So negative charges on both sides, they repel each other. Cell is not stained, only the background. These stains are convenient to visualize details of the outside, like, you know, the details of the cell envelope, right? Outer cell structure. Differential stains, they can be for different purposes. One of the most fundamental stains is the Gram stain. You can see the example of Gram stain on this micrograph. The purple cocci are the gram-positive Staph aureus. The red rods, the quality of the micrograph does not let us appreciate that they are rods, but trust me, they are. It's E. coli, gram-negative. So you have some bacteria that appear blue to the Gram stain, and some bacteria that appear red under the Gram stain. Make sense so far? Why it's so important? Why being Gram? So does it reflect something? Being Gram positive or Gram negative, or it's just some fancy feature? The answer is yes. Gram positive and Gram negative bacteria are fundamentally different in terms of the structure of the cell wall. And that has consequences like resistance to antibiotics, ability to cause various inflammatory conditions and various symptoms in bacterial disease, ability to resist cleaning agents, okay? So I don't really know when Hans Christian Gram, the guy who actually discovered that staining, I don't think he knew about the differences in a cell wall structure, but it turns out he found some very fundamental feature, okay? There are also acid-fast staining, which allows you to distinguish specifically mycobacteria, like the causative agent of tuberculosis. Staining for capsules, protective coating of bacterial cells, and staining for flagella. Okay. Now, basic stains are usually associated with a positive staining, and acidic stains are usually associated with negative. So I mean, most of the negative stains are acidic stains. Most of the basic stains are positive stains. Any questions on those categories? So we have simple versus differential. Simple don't distinguish different species. Differential can. Now let's chat about one of the most important differential stain that we will do in the class. Not today, but we will. So this is Gram stain, and it starts with staining your sample with crystal violet. I just mentioned it several times. Crystal violet, is that simple or differential stain by itself? It's simple. All cells will appear blue. Gram positive and Gram negative cells will appear blue. Next, you add iodine. So what iodine does, it makes crystal violet to better stick to the cells. You follow me? It's called mordant. Cells don't change appearance actually. They're still blue. What you do next is you wash your sample with a little bit of alcohol. So alcohol will remove crystal violet. Turns out that crystal violet, after treatment with iodine, sticks better to gram-positive than to gram-negative. So alcohol will not be able to wash away crystal violet from gram-positive cells. They will remain blue. But it will wash away crystal violet from gram-negatives. So they will become discolorized. They will lose color. Alright? The last step is to give gram-negatives a color. For this we add safranin. Safranin gives these cells red color. That process makes sense. Actually, unless you do it, it does not, honestly. And unless you do it like 2-3 times, it doesn't make much sense. But when we will do it, it will start making sense. This is one of the keystone techniques that you absolutely must be familiar with. Not for the purposes of my course, not for the purposes of graduating from Lakeland Community College, but this is something that you'll run into in the clinical lab. I listened to a podcast. They had an interview with the chief of the clinical lab operations at Stanford University School of Medicine, Health Center, and stuff like that. Okay, hospitals, Stanford University hospitals, like a big thing. They have to carry some of the samples from one hospital to the lab, like they use truck because it's so far. Okay, and this lady was telling about new techniques in diagnostics of bacterial and viral diseases and blah blah blah, and it's very interesting. But then the host asks her, so what is the first to go technique in identification of bacterial samples? And she answers just like this: gram staining. So you have physician supplies the sample, they put it on the plate, they grow microbes, they stain it. First thing, with all the fancy stuff, you have to stain it. So you have to know that. Even if you don't do it, when people from the lab call you and tell you, it's gram-positive, you know what they're talking about. Make sense? Okay. Other stains that are differential stains. Acidfast. Mycobacteria, like tuberculosis or leprae. Mycobacteria leprae is a causative agent of leprosy. They're slightly different in terms of the cell wall structure. So we use the special stain named Acidfast. The difference is different dyes. Again, cells like mycobacteria, they retain special dye after treatment with a special decolorizing solution. Okay? You can see them stained red here. Okay? Some stains allow you to stain for capsule. We have capsule, yes. So capsular staining. Capsule is a protective coating around the cell. It protects cells from phagocytosis, from response of your own immune system. Now, if bacteria have capsule, are they going to be more pathogenic or less pathogenic than non-encapsulated? What do you think? If capsule protects them from immune system, more, of course. In this case, capsule can be considered as the virulence factor. Okay? It's important to know whether cells have or don't have capsule. You see the logic? Okay. Endospore staining. Certain cells, certain bacterial species, can make endospores. Endospores have nothing to do with fungal spores that you can see, and you kick the mushroom and there's a cloud of spores. It's a different story. Endospores allow bacterial cells to survive through harsh conditions. And one thing that bacterial cells absolutely hate is dry environment, desiccation. Remember when we examined your hand wash samples? I think some of you had like these big, gigantic colonies, very odd looking. You remember? No? Usually it was like smaller white, smaller yellow, but in a couple of plates there were like big colonies. That was bacillus. What happened is you collected the endospore from somewhere, touching something dusty, dry and dusty. And when you place this endospore on the plate, endospore being in a wet, nutrient-rich environment, germinated and bacteria kind of popped up, like a bird from an egg, okay, something like that. So knowing if you have a spore former, like this Bacillus subtilis, can help you in diagnosing the disease. Flagella. What is the problem with visualizing the appendage, cellular appendage, like a flagella? It's too thin, right? You're not going to see it. It's too small. So if it's too small, how can you make it visible under the light microscope? What does it do to flagella if it's too small? It makes it big. Yeah, it sticks to it. And flagella, from being really thin, becomes really thick, like gigantic. Okay? So in real life, flagella is not that thick. You see how big they are? Oh no, they're much thinner. Just staining that gives them this appearance. Why flagellar staining? What is the purpose? I mean, obviously, the purpose of flagellar staining is to see flagella. What is the relevance? What is the importance of seeing flagella or not? What's the function of it? Mobility. Very good. So cell, if you see flagella, it means cells, these bacteria, can move. Which bacteria potentially pose more serious threat? The ones that can move, or the ones that are passively carried through the body by bodily fluids? Yeah, ones that can move. They're usually more invasive. They can swim to different places, right? So that gives you a link to the clinical aspects. I'm not going to go through the summary. This table is for your reference, okay? You will see pretty much the same pictures, all the same pictures, except for the capsule stain. For capsule, the picture is different, okay? So now, you may ask, oh, good God, what do we have to know about the staining? About this various methods? Well, you have to understand the idea of simple versus differential. That's one. You have to understand the idea of positive versus negative staining. Which one will stain cells, which one will stain the background. You have to understand those, what did we count? 4, 5 basic types of differential staining. Gram, acid-fast, capsular, endospore, and flagellar. So 5 types. one of the questions that usually arises is, do we have to know the exact names of reagents? My answer is no. So you don't have to know if I ask you, which of the following dyes is used for capsule staining? You have to pick, say, India ink. God forbid, no. But I can give you an actual example. Such and such dye penetrates and is retained by the walls of Mycobacteria tuberculosis. Okay? Would it be example of gram staining, Acidfast, so on? So you have to know the purpose of the staining rather than all those chemicals. Did I make myself clear about that? Okay. Any questions on the staining? Okay. Let's move on. So next topic, very interesting topic that we're going to chat about is how we culture the microbes. So why do we need to do that? Why do we need to culture them? What's the purpose of culturing microbes? In what sense? That's right, yeah, better sample. In what sense? So we can see them, right? Maybe that's, we don't have enough, so we need to have more. Something else, yes. Wait. Keep it. I think I know what you mean deep inside. When we, yes. Reproduce faster, yes. So we can accumulate. Actually, the next question would be, I don't remember if we talked about it or not. Microbes, where would they grow faster? On the guard plate or in your stomach? What do you think? You think stomach? Who thinks a guard plate? Let's vote. Voting doesn't solve problems, it rather creates them, but let's vote. Who's for the stomach? 1, 2, 3, 4, 5, 6. And how many people? Well, I suppose everybody else is for a guard plate. And we have a guard plate. A guard plate wins. Now, a guard plate is a correct answer. Now, knowing that the same microbe will grow faster in the agar plate than in the stomach or the intestine, can you explain it knowing that? What's the main difference between a agar plate and the stomach? Expand on it. Some guys prefer acid. You can create acid. You can create the necessary environment, controlled. You close, not yeah, it's kind of a safe, right? What about nutrient availability? Like as much as they can eat, technically speaking. I mean, unless they grow like over the whole plate, and where the competition is tougher. In the stomach, of course, because it's not only one species or one microbe, okay? It's hundreds and thousands of them. Does that make sense? If actually I had to read about it recently, I dug into it. The difference in generation time for E. coli on the plate, generation time is about 20 minutes. In the intestines, it depends on the study, roughly about 2 hours. It's considerable difference, you know. So put them on the plate so they can grow faster. What if you have several species in a sample? Can you now detect all of them, see all of them? Yeah. Like we saw different types of colonies in our hand wash plates, right? And when you grow a lot of them, you can put them under the microscope. You can subject those samples to other methods of studying, essentially identifying what the heck is going on in the clinical sample that you are working with. So culturing microbes incorporate 4 or 5 I's. You inoculate the medium first, incubate it, isolate whatever you want to isolate, and then inspect your results and identify whatever you want to identify. And we're going to talk about each of these aspects, each of these steps separately. So first is inoculation. When you did this with a cotton swab, what did you do essentially? What did you collect? A sample, yeah. So collecting a sample is first step of inoculation. Since we talk mostly about clinical aspects, say you're swabbing, or you're collecting somebody's blood, or you're collecting somebody's urine. What is the common condition for all these 3 methods? Yes, it should be done in a sterile fashion, in sterile, well, not sterile environment, but syringe must be sterile, the urine collection cup must be sterile, cotton swab must be sterile. We got it? What can serve as the clinical sample? Pretty much anything, yeah. So you can collect, help me, body. We can, if you want, we can expand blood, urine. Huh? You can collect stool, mucus. You can collect saliva, right? In some cases, people collect lacrimal secretions, tears. You can collect CSF, cerebrospinal fluid. You can collect gastric juice. You can collect biopsies. You name it. Really, you can collect virtually. Now, this is something, again, very important. You have to think what you are collecting. One of the best examples, have you heard about intestinal microbiome? Well, I bet you did, right? About 20 to 30% of your stool weight are microbial cells. So, feces, right? There are a lot of microbes in there. You can take the sample, fecal sample, put it on the plate. A lot of them will grow. Make sense? Feces. Is that intestinal microbiota? Not really. Here's the thing. We have microbes in the intestines, 1,500 species, probably more than that. We know about 1,500 species. And then you defecate. Well, some of them will go with the stool better than others. Does that make sense? So actual ratio of different species in the intestine is not necessarily reflected by whatever you find in feces. And it was shown that if you compare samples and the samples that were obtained as a result of colonoscopy and samples of feces, they are not matching 100%. So ideally, we would actually go through people and just do colonoscopy to them. I don't think a lot of people would agree to be a subject for colonoscopy on a regular basis for scientific reason, only scientific reason, without any health reasons. So it's kind of what they do now actually. And colonoscopy is, and you know, I wouldn't agree to, well, probably I would. If somebody would do colonoscopy on me, I would say, yeah, take a biopsy of the intestine anyway, but not everybody would do that. So what they do now, when they do colonoscopy, there's a device that allows you to wash the intestine and collect that wash with intestinal microbes. It's pretty fancy. Well, the reason I know so much about this, my wife works in the lab, which is a part of gastroenterology department at Case. So I spoke with her boss a lot, and he enlightened me about process of colonoscopy. So that's clinical samples. A lot of things that are relevant for your future profession. Environmental samples, although you're probably not going to be collecting them, are no less interesting. Some of them are very simple. Soil, you go and collect. Water, you go and collect. Actually going to be collecting some water that semester, and we're going to test it for some stuff. Okay. Now, there's a lady. I have horrible memory for names, so don't ask me what's her name. I can look it up. And she's either in Akron or Kent State, somewhere there south. She studies microbes of the caves. So she goes into the cave and collects microbial samples and studies microbial communities. It may sound pretty dull. I mean, except for going into a cave, it's kind of fun, right? Think about this. Cave must be normally inaccessible to humans. Otherwise, what you're going to find there is human stuff. Second, when humans go there, they should leave no microbial traces, which means protective gowns all the time. And again, I get a lot of stuff from podcasts, but when she was talking about it, she said, and we have to collect all our waste. Initially, I thought about garbage. No, we talk about human waste. Urine and stool have to be collected and transported out of the cave. You can't dump stuff there, so you don't disturb the microbiome. It's pretty interesting. Okay. So you've collected environmental samples. We're actually going to be testing some food. That becomes one of the most exciting labs that we do. And then once you collected the sample, you have to inoculate the medium. What can serve as the medium? You can look at the slide, but also give me some other examples. What can serve as the medium? Yeah, you can inoculate blood, absolutely. You can cultivate your microbes in blood. Something else. What did you grow your samples on? Huh? Agar, yes. Special agar. You can use it, right, as a medium. What happens to the milk if you leave it out of the fridge for 2-3 days? Why? Why it goes bad? Yeah, bacteria. It's not sterile, it's just pasteurized. So bacteria make it bad. The milk is the medium in this case. Same goes for, say, chicken broth, okay, or beef stock that you cook and accidentally forget. That's the medium. Anything that supports growth of bacteria is medium. I mean, this table is not the medium. Why? Why it does not support the bacterial growth? Huh?
It's dry.
Excellent. First of all, it is dry. And what else? Yeah. So it's no nutrients, is eutrophic, and a little nutrients is oligotrophic. Does that make sense? Right? It just names. So trophic means tropho, food. Okay? Oligo, a little U, no. Okay? You can actually see it sometimes in ponds when they have a massive algal bloom, like massive. Well, algae is slightly different, but sometimes with fish it can happen. When fish eats out all the nutritional sources, and they will die because they make the pond eutrophic for their species. So this is eutrophic environment. Anything with just tad food is oligotrophic. Our skin is oligotrophic, actually. There's very limited amount of nutrients available. Okay? So you see examples of medium. The agar plate, the blood, eggs. You can grow microbes in animals. You can grow microbes on plants. You can grow microbes on cells. Okay? Media is not only necessary to grow organisms. It helps you, in some cases, to identify them. Because some media will support growth of this kind, but not of this kind. They may help you to differentiate microorganisms. Does that make sense? Okay. And we're going to talk about them. Basic classifications of media are based on the physical state, based on what's in the medium, and what is the purpose of the media. Why do we use it? Okay? And in the following probably 15 minutes, I hope we will discuss those different types. Based on the physical state, you've all seen agar plates, well, I suppose. Agar is solid, right? It's pretty obvious. Solid and liquid. There's liquid medium here with the microbe in it. Okay. Now, what are the advantages, disadvantages? Well, solid is fairly easy to handle. You can see the solid medium here. Nobody uses it like that. Really. No. Well, I can imagine in some cases, but it's not very common. Mostly solid medium is used in the form of the petri dishes with agar on it. Right? It allows you to grow microbes in the form of colonies. And that's a unique feature of the solid medium, because you can characterize your microbe based on the colony shape, size, or color, as you just did with your hand washing. How many types of colonies we were able to identify? Mainly two, white and yellow. In some cases we had this big yellow, big white, right? Huh? And you had red one, yes. So actually it comes down to four. Okay, 4 different types of colonies. And they were quite different, right? How do we put this solid medium together? I can tell you. With a big flask, glass flask. And we throw all the components in the flask. We pour water, stick it in the autoclave. Autoclave is right behind that wall. I mean, you're not going to see it, but if you want, I can give you a little tour of the prep room. It's pretty boring, except even autoclave is boring. It's like a giant box with a heavy door. That's it. It's like pressure cooker. Just very, very big pressure cooker. Okay. So stick the stuff into it, kind of boil it, sterilize it, take it out. It's all liquid. Agar, when you heat it up, becomes liquid in water. Okay. And you pour it in the plates, leave them out on the table for a couple of hours. They cool down. As it cools down, it solidifies. There you have your agar. So like, if we take those plates, throw them in the autoclave, agar will melt. Okay? Something like this you can do at home. Any ideas? Not with agar. Well, butter, yes, I never thought about it. It's actually a great example. You can liquefy butter and it will solidify back. It won't be a great medium because it does not, or the basis of the medium. Probably not. Because it's just fat. It's so much fat. Okay, yes, you can do that. Streaking it is going to be a bit of a problem. Honey, when it becomes solid, it because sugar crystals become larger. Jello. You can do Jello at home, right? It is liquid and then you leave it to cool down and it becomes solid. But there is a reason why we don't use gelatin as the basis for medium. Bacteria eat it. It would be a bit of a disappointment when you put bacteria in a gelatin plate, you come back later in the day, and everything's gone. Bacteria is dead. Gelatin's eaten. Bacteria is dead. Your sample's over. Okay, it's kind of a bummer. Probably so you can see how they digest. Yep. Or you can take someone with necrotizing fasciitis and just do a time lapse of how flesh is melted off the bones. It's even more exciting. Not humane, but exciting. Unfortunately, there are plenty. Not volunteers, but people with, more than you think. Whatever they call flesh-eating bacteria, it happens pretty frequently. Now, liquid medium, opposite. Here, obviously you can't isolate any colonies. Okay. You can make it at home, as we mentioned. Chicken broth is the perfect liquid medium, especially if it's transparent, so you can see bacterial growth. Does that make sense? So if you can just cook the chicken broth and filter it really well so it becomes not turbid, and you can see how bacteria grow. Seriously, you can do that. Okay? The downside, you can separate species. So you have a growth and you don't know how many different types are there, right? The upside, you can add indicators. Like in this picture. When you inoculate these tubes, the bacteria that break down urea, ammonia, which is the product of that breakdown, changes the pH of the medium, turning it pink. Okay. Now, in between is semi-solid medium. Semi-solid is, we call it a clot, and we're going to use it at some point. Semi-solid medium, if you just hold it or just turn it sideways, it will not leak out of the tube. But if you turn it upside down, if you turn the tube upside down, okay, eventually it will kind of slide away. Does that make sense? There's no solid surface of it. Okay, why do we need that? Huh? That belongs to the microscopy realm. It just, I see what you're saying. Oil behaves differently from water. It's more dense, it's more viscous, but it's not semi-solid. Take oil. Do I have the oil? I bet somebody stole it from me. Yeah, somebody stole it from here. But if you take the immersion oil, yeah, It is viscous, but it's fluid. Okay, so I see how it, the only problem is that only I can see how it's about to spill out of the bottle. Okay, this one will not. I mean, I can turn it out. Nothing's going to happen in at least first 3, 4, 5 minutes. Okay. Now this medium is used to test the ability of microbes to move. When medium is solid, it's not really easy. Okay, when medium is semi-solid, they can move. When medium is liquid, they all just move around, so you can't really distinguish the ones that can and the ones that cannot move. Does that make sense? So in semi-solid medium, look, this is uninoculated sample, right? Now, when we stick something that cannot move, we take like a needle. This is not a needle, but imagine it is. And pierce the medium through. What happens? Microbes that cannot move stay at the site of inoculation. See that? That trace here? Okay? Now microbes that can move will leave no trace, but instead they will make the whole medium turbid, cloudy. They will spread through the whole clot. Make sense? So here you see motile and non-motile. You may ask, what the fuck is that? It's black. Now, this is something that you can do with the medium. You can combine different qualities that will let you to characterize microbes from different aspects. So here you see, alright, this is the motile dude. This black comes from the microbe that reduces sulfur. So this medium, when sulfur reducing microbes grow in it, they go from this to this. So this is reduced form of sulfur. This is sulfide ion. When sulfide ion binds to iron, they form, iron 2, sulfur 3, iron sulfide. It precipitates immediately. It's not dissolvable in water. It precipitates and it's pitch black. So here you can say this is the motile microbe that also reduces sulfur. Now, why there's so much buzz about reducing sulfur? Different species, different genera of microbes can be differentiated based on their biochemical characteristics. Ability to reduce sulfur is just a feature. Does that make sense? So you can say this is some sort of bacteria, okay? Composition. Media can be synthetic, can be complex. Synthetic means that you know its chemical composition. You can see this is the chemically defined or synthetic medium for cultivating Staphylococcus aureus. You can trace the exact amount of each compound from amino acids to vitamins to salts. Pretty much you walk in the lab, you have shelves with chemicals and you put them in a flask, you add water, you autoclave it, here's your defined medium. Make sense? Okay. Complex. Something is not chemically defined, like brain-heart infusion. Brain and heart tissue is lyophilized, turned into powder. When you need medium, you just throw, pretty much like make a solution of brain and heart. Your beef stock or chicken broth is complex medium. Do you know the exact chemical composition of chicken broth that you cook it out? No, you don't. Do you know the exact chemical composition of that gallon of milk that you take home from the grocery store? No, you don't. But you don't really care, because what you need complex medium for is growth of whatever, right? That TSA plates, the TSA dishes, the Petri dishes that we used. TSA stands for trypticase soy agar. So it's an extract of proteins from soybeans. And those proteins are partially digested by the trypsin. There's no way we know chemical composition of it. But we know that there is a lot of stuff in that medium which will support microbes with different nutritional requirements. Does that make sense? Think about this. Complex medium is your buffet. Okay? You don't know what's in the buffet, but you're pretty sure you're going to find something that you can eat. Okay? Synthetic medium is like going to your friend. You know that there's going to be only chips and salsa, and that's it. Nothing else. But you know for sure that's what's going to be there. Okay? And if you can't survive on chips and salsa, well, too bad for you. Now, if we take a look at this media, complex, which support growth of a bunch of microbes, and defined, which corresponds to exact nutritional needs of a particular organism. The question that may arise is, why do we use synthetic medium? What is the point? Why don't we just skip it and just grow microbes on the complex medium? Which advantage can synthetic medium give us? Excellent. We can manipulate it. How? What can we do with medium which chemical composition we know exactly? Huh? Say again? To the microbe. But what can we do with those chemicals? Yes. In the controlled manner. We can take out glycine and tell the microbe, well, quote unquote, tell the microbe, now grow. Does that make sense? Say, we want to select for mutants that can survive without lysine. We take out lysine and we grow microbes on that medium. Does that make sense? We can manipulate it, okay? We can change it because we know the exact nutritional requirements for a particular organism. When we were collecting samples from our hands, we didn't know what's going to be there, right? Since we don't know, we want to provide as many options to those microbes as we can. Agreed? Okay. Let's take a break before we get to the different